Welcome to the first technical session of today's national webinar. It is a pleasure to have with us an experienced personality, Dr. Tathagata Dutta sir, President and Chief Technical Officer, Jodas Expoim Private Limited, Hyderabad. Dr. Tathagata Dutta sir holds a B.Pharm, M.Pharm and PhD in Pharmaceutical Sciences from Dr. Hari Singh Gaur University, Sagar. He further pursued postdoctoral studies at the School of Pharmacy and Diamantina Institute for Cancer, Immunology and Metabolic Medicine, University of Queensland, Brisbane, Australia, focusing on the development of RNAi therapeutics for cancer management. Additionally, he is an alumnus of IIM Calcutta. Over the span of more than 2 decades, Dr. Datta has held various esteemed positions, including President, Chief Scientific Officer, Vice President, Executive Director, Managing Director, and CEO within several reputable multinational pharmaceutical companies worldwide. His contributions to the field have been recognized with numerous national and international awards and honors. Dr. Datta's research endeavors have led to the publication of several papers in renowned international journals, human patents, and authored book chapters. He serves as an editorial board member of several international journals focusing on drug discovery and delivery research. His research interests span a wide array of topics, including novel drug delivery system, dendrimers, liposomes, polymers, biconjugates, tissue-targeted drug delivery system, nano-drug delivery system, gene therapy, vaccine, genetic immunization, depot injections, gene delivery, and the development of RNAi therapeutics. Now, I would like to request Sir to please address the gathering. Please, Sir. I would like to request Sir to please address the gathering.
Just wait for a minute. Dr. Datta is starting the session soon.
Okay, sir. Thank you.
Let me thank Dr. Datta. In spite of his busy schedule, and right now he's not in India. He is traveling. He is in Italy. But since we requested to take the session, so it's too early in the Italy, but he is joining soon. I think within a minute, he will start the session.
Okay, sir. No problem.
Yeah.
एक मिनट में आ
Hello.
Hello, sir.
Yes, madam. Yes,
Yes,
Dr.
sir, you're
Datta.
audible.
Yeah, am I audible and visible? Yes,
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 sir.
Dr. Datta. Okay. Dr. Mishra, so shall I start? Yes,
Yeah,
please.
you can.
Okay, just a minute.
Wonderful to see
Let me.
you, Dr. Datta. Wonderful.
Thank you, sir. Thank you so much. It's a pleasure. Uh, so very good afternoon, everyone. It is my pleasure to present before you probably first time before the association gathering regarding long-acting injectables for effective management of reproductive tract diseases. So those who are from pharmacy background, I think they will really enjoy this because I am from industry and these are something which we do, and those who are the clinicians, they can correlate their experience with what I will be talking, because the things which we are developing or we produced, they are in market, they can correlate their practical experience with the formulation aspect, and they will be able to enjoy it as well. I will be speaking very slowly, but I would prefer if people can interrupt me in between if there is any question and ask me. So this is just a very formal disclaimer. So the outline talk about long-acting injection, long-acting suspensions, how the formulations are developed, microspheres, nanoparticles, characterization, how are done, critical parameters, process challenges, how in vitro release testing and drug absorption and bioavailability, and how they are related to reproductive tract diseases. So you can see most of them are not related to clinical things, but yes, the entire presentation will be based on clinical aspects only. And at present, whatever the long-acting injections are there in the market, 90% of the drugs are meant for treatment of reproductive tract diseases. And they are mostly peptides or steroids. And these long-acting injections are like wonder drugs, because for treatment of any reproductive diseases, whether it is a steroid or a hormone or some hormone analogs that are agonist, antagonist, that are given via injectable route. The treatment is actually for a prolonged period. It cannot be something like you take a course of an antibiotic for 5 days and you are cured of the disease, or you can continue it for say 7 days or 2 days. It's no, like normally for skin infection, steroids are given for 5 days or 7 days therapy. It is not like that. The therapy continues for minimum 3 months, 6 months, sometimes years together. And there we have to keep in mind the patient compliance, the ease of administration, and obviously when you take such an agent, the fluctuation in the dosage form, that is the crest and troughs. So when the
plasma drug concentration is very high, then there are serious adverse effects. And when the plasma drug concentration drops, then there are again lack of efficacy. So we need a steady plasma drug concentration, and which we get a very good therapeutic response. So there, these long-acting injections comes into play. And very classic, very primitive form of this nanotechnology-based long-acting injections are obviously suspensions, nano suspensions. So and then obviously further they comes in the form of microspheres, nanoparticles, which are a bit more expensive and more technology oriented. So they are controlled release injections which provide controlled release of the drug for a prolonged period of time, which might extend from several days to several months when administered through intramuscular, subcutaneous, epidural, or intra-articular route. Here it is written several months, but yes, we know there are now long-acting injections which are meant for years. And obviously, there is another category of finished doses which the clinicians are very well aware of. They are called implants, subcutaneous implants. So where the small incision is made in the skin and these implants are inserted and stitched, and that implant releases the drug for a year or so. So these are all nothing but long-acting injections. It's only the difference in terminology. So here I will be talking about suspension for injections, microspheres, and something which is very close to my heart, and I have certain patented products on that as well. These are in situ gel forming implants. So as I said, when injecting a suspension or a microsphere or a nanoparticle is a very painful job. So typically a 20 gauge needle or sometimes 18 gauge needle is also required to inject these long-acting injections. Obviously, at the site of injection, there will be a pain as long as the depot is there, as long as the depot for this long-acting injection is there within the site. But there are a category of injectables which are called in situ gel forming implants. These are nothing but injections where these are liquid injections, and when they are injected, polymer-based injections, they have ease of administration. They are since they are liquid in nature, low viscosity, they have very good syringeability and injectability. And once they are injected, they come in contact at the physiological pH. They get converted into a gel. They form a depot in situ inside the body, and they releases the drug at a controlled rate for months or even years. The development of long-acting injections is a highly challenging task, be it the formulation, the analytical, the bioequivalence study, or the clinical part. There are several controversies associated with the long-acting injections. Them more often, injection visit visit to the clinic will not be there. So this is something, the irony. That's why in the obvious associated with longer time. So there are because of two reasons we see that countries where the healthcare system is not very well developed, there the long-acting injections for all these reproductive tract or organ-related diseases are not very popular. There, the doctors prefer that the patient will visit them once a day and they will take the shot, or they will visit once a week or take the shot, or even they will take the tablets. So these are, then there are several, there's sometimes suspension of crystalline drugs in prefilled syringes. The powder intended to be mixed with required media for suspension or solution Usually solution for injections can also be long-acting injection, but to make a solution for injection, long-acting injection is the classical way is all these your drugs, such drugs are usually lipid soluble. That means fat soluble. They are not insoluble. They are insoluble in water. So if you can mix them in oil and inject it, then they become inherently long acting. So do we have such a solution for injections also long acting? And when you make this insoluble things as a suspension, nano suspension, and disperse it in aqueous media, they again become long acting. These are the two very simplest form of long acting injection. One long-acting injection, which I think all of us in our common day-to-day life have come across is vitamin D3 long-acting injection. So vitamin D3 is nothing but a oil. Most of the world population suffers from vitamin D3 deficiency. And because it is available in the form of either soft gelatin capsules or suspensions for oral suspensions or something, their bioavailability is very, very poor. And because of that, even people will take oral route of this vitamin D3, they will be having vitamin D3 deficiencies and symptoms associated with it, like back pain or pain in the muscles and sometimes depression, all these things. But if you take, there is a long, many people, I don't know whether they are aware or not, there is a long acting injection called Arachidol by Abbott. And this injection, when given into preferably to the buttocks, this injection, there are in two strengths, 3 lakh international unit and 6 lakhs international unit. So if you give a 3 lakh international unit, you will have a constant vitamin D3 concentration for 3 months. 6 lakhs international unit means complete for 1 year, you will have a continuous steady vitamin D3 concentration in the body. But it is just a solution of vitamin D3 in cottonseed oil. Suspensions, where you suspend the drug, and this suspension, when you inject it, they form the depot. Coming to this reproductive tract disorders, most common long-acting injection, which is a suspension dosage form, is medroxyprogesterone acetate, which commonly in US it is known as Depo-Provera. So this is actually medroxyprogesterone, and it releases the drug at a sustained rate for a prolonged period. These drug particles are in the form of micro or nanocrystals. The nanocrystals of long-acting injections usually follows Noyes-Whitney equation for drug release. So those who are from the pharmaceutics background, they know that there are different equations for drug release, like so Noyes-Whitney equation is there, Higuchi is there, and all this there. So mostly the polymers which we use for preparing these long-acting injections are PLA and PLGA based. PLA means polylactic acid and PLGA means polylactic D-glycolactic acid, copolymer. These are suitable for the delivery of peptides and proteins. So again, when we talk about the drugs that are for disorders like reproductive tract disorders, drugs like leuprolide acetate or triptorelin, all these drugs are given as a long-acting injection in the form of PLA, PLGA-based microspheres. So these PLA, PLGA-based microspheres of triptorelin, leuprolide, all these are nothing but peptides. Here quickly, I want to add a regulatory perspective to it. So what are peptides? What are proteins? Usually, common language, we say long chain amino, any molecule which contains more than, say, 10 or 12 amide linkages are considered as proteins, and remaining are all are peptides, dipeptide, tetrapeptide, decapeptide, all these things. But yes, US FDA has a specific guideline to that, which says that any molecule that had more than 40 peptide linkage is considered as a, or 40 amino acid is considered as a protein, and less than 40 is considered as a peptide. So all these things are all peptide, comes under the category of peptides and not proteins. The route of administration is very, very important, and that will determine the safety and efficacy of this long-acting injection of the drugs used for reproductive tract disorders. Mostly these are given as a deep intramuscular injection, preferably in the buttocks. So I will show with data, some of the data I will show that how the site of administration or the angle of the syringe or the needle, the drug polymer ratio, the degradation rate of the polymer, the particle size of the microsphere, polymer oligomer monomer ratio, all these factors, how it actually interferes or decides the safety and efficacy of the formulation. Glass transition temperature, monomer ratio, amorphous versus crystallinity, molecular weight, intrinsic viscosity, solubility, and yes, very important thing is end group, that is ester versus acid group. Stabilizers, whether we have used or not, organic solvents, temperature, solvent evaporation rate, lyophilization cycle, and zeta potential, pressure, agitation rate, geometry of reactor agitator, surface morphology, shape, and particle size. So there are typically, we prepare these long-acting injections by techniques like coagurbation phase separation, emulsification solvent evaporation, and spray drying. So I had forgotten to tell you, whatever I present, wherever I present, I make my presentation, though I am from industry, I make my presentation freely available to the entire audience. So immediately after this talk is over, I'll share a PDF copy of this presentation to the organizers, and whoever is interested, they can get their copy from the soft copy from the organizers. And anybody having any further questions also, they can contact me over mail or WhatsApp later stage also. My only humble request is, since I am from industry, whatever images I am showing in this presentation, all are copyright protected, all are intellectual properties. So please don't reproduce them. You can reproduce the knowledge, you can reproduce the text material, obviously, I don't have any problem. But don't use the images, scanning electron microscopy, transmission electron microscopy and all, because most of them are submitted to different regulatory agencies. So content uniformity. So this is one problem which we face when we prepare this long-acting injection. So one vial to vial, the dosage units, the variation is there. We have to be very, very critical about that. And the polydispersity index plays a major role in that. Same is with aggregation. If there is an aggregation, then the amount of the drug loaded will be more in a particular prefilled syringe or a vial, and the patient will get a higher dose. Residual solvents are toxic in nature, and mostly we use organic solvents like chloroform or dichloromethane to prepare these long-acting injections, which are the US FDA limit is very, very stringent. That is around 60 parts per million. So obviously that is there. Development of static charge that prevents in the formulation to have the desired release profile at every stage, choking and loss of potency. We do the characterization of these long-acting injections by different techniques like gel permission chromatography, nuclear magnetic resonance chromatography, particle size distribution, differential scanning colorimetry, scanning electron microscopy, and transmission electron microscopy. So scanning electron microscopy is done to check the surface morphology, topography, while transmission electron microscopy is done to go through the internal structures of the dosage form. SDS paste, that is sodium dodecyl sulfate, polyacrylamide gel electrophoresis. This is used and the easiest technique to check the impurity profile of the formulation, because the API, which are basically peptides in nature, they usually break down and those gives multiple bands. So this is the quickest way we can check how much impurity is increasing. And the most critical thing is the in vitro drug release. That is the drug release profile of the formulation inside the body, because if it releases the drug at a faster rate, then the dose dumping will happen, and if there is a dose dumping, then the patient will have serious adverse effects. On the other hand, if the release profile is slow, then that concentration of the drug in the plasma will be less than the minimum effective concentration, and therefore we will not get any effective therapeutic concentration at all. We will not get any efficacy. So we have to have the ideal drug release profile, and that should be typically a zero order release, but there are only very few doses from which can give a zero order release. So it is almost impossible. We get something around pseudo zero order release. So the type for apparatus, the apparatuses which we use for this type of drug release profile testing are USP type 2 with dialysis sac. That is, either you have a paddle or you have tie with the paddle a dialysis sac, and you fill the microsphere or the suspension or the oil inside that, and you check it. Use USP type 2 also with enhancer cells, or you use USP type 4, that is flow through cell with semi-solid adapters, and this is usually we use a closed loop system for this. Now here, some of the guidelines which are provided, though these are not meant for the long-acting injections for reproductive tract disorder. This figure only contains some of the antipsychotic drugs, except obviously methylprednisolone, which is a steroid. But mechanism is all same. So you can see that what the US FDA has proposed, the in vitro drug release profile. So in vitro drug release profile will be something which will mimic the exact condition it will face, the injection will face when administered to the body. And you can see, like, say 480 minutes up to 480 minutes, we do that release profile. Sometimes we do 360 minutes. Now, one question I frequently encounter from people is, sir, you are saying that this drug release profile is up to the drug will be once injected will exert its efficacy up to 6 months or 1 year, and you are doing a release testing up to 480 hours. Why? So see, to check the release profile, in vitro drug release profile outside the body for more than, say, for months or years is practically not possible. That is first thing. And second thing is, what is our aim? Our aim is initial drug release will start from the delivery system, and it will, at certain stage, it will reach the peak concentration, Cmax, peak plasma concentration. And after that, it will reach a concentration which we call as CSS, steady state plasma concentration. Once the drug reaches steady state plasma concentration, we will be terminating the study, because we know now I have received the desired concentration, and it will continue for the rest of the thing. And also when it reaches the steady state plasma drug concentration, it is practically now predictable that after how many days, months, the drug concentration will again start declining, or again it will come down to subtherapeutic range where a second dose will be required. See, here you have methylprednisolone, paliperidone palmitate, olanzapine palmitate. So here you can see the scanning electron microscopy images of leprolide acetate microspheres. Leprolide is acetate, you know, is a peptide, and this is given as a liquid injection also, immediate release, as well as long-acting injection in the form of microspheres, which are available 1 month, 3 month depot injections. Now, there are many brands available in the domestic market. Once we, since I never worked for domestic market, but one of my relative was suffering from endometriosis, and she was recommended to be administered leprolide acetate long-acting injection, this microspheres. She was taking this injection, but without any relief. So once when I saw the injection, it was some XYZ pharma company in India. Once I saw the injection, I suspected that whether it is really a long-acting injection or not, because I can only see powders inside it, not any granular microspheres. So I did some characterization study and found out that there are only drug powders are there. There is no microsphere is there. There is no PLA, no PLG. That is the reason she was getting the dose dumping. She was having serious adverse effects, nothing much. That was the time I purchased 2 lots of two injections of the same from US, manufactured by a very, very reputed Indian pharma company in Gujarat, based out of Gujarat. But I bought the injection from US and I gave it to her. And trust me, after that 2 injections, she was completely recovered. And this was so the particle size and the entrapment of the drug in this is very, very important. So in this image, you can see some of the surface is rough. Sometimes some drug is adhering to the surface. This actually will give burst release, initial burst release, and will lead to toxicity. And again, these are of 2 types. Again, we call it, one is porous and another one is non-porous. So this is the porous one, microsphere, and these are the porous microspheres. You can see, so the porosity are very, very high. So how porosity appears in the microspheres are prepared by different techniques which involve solvents, and later on when you remove the solvent, if the solvent evaporation takes place at a very fast rate, then the porosity becomes high and the drug release happens very fast. The dose dumping happens. If the microspheres are dried very, very slowly, like over a period of 7 days or 10 days, then the microsphere surface becomes very, very smooth without any micropores. The pores will be in the size of nano or picometer, and the release profile will be very, very constant. Efficacy will be very high. Safety will be very low. Here you can see that the site of injection, sex, physical activity, disease state, pH, osmolarity, and excipient used. So till now I was talking of something. So I don't know what is the composition of the audience, but I felt most of you are from clinical background. So I skipped the earlier slides at a very fast pace because I felt these are not very much interesting to people like you. But the things which I am going to talk now is going to be very, very interesting for the clinicians, the doctors, the nurses, and some of the pharmacists, hospital pharmacists as well. And they can correlate their experience with this. So most important thing is now the site of injection. So you can see in the figure, what are the sites of injection? Either you can give it in the gluteus muscle or you can give it in the deltoid muscle. Wherever you give it, first thing you can see from in the anti-clockwise direction, if you see the second image at the bottom, you can see that the syringe, the position of the syringe in two different ways. One is that is 90-degree angle. Another one syringe will be something like some people keep it 60-degree or 30-degree angle. So if you see, if the position of the syringe is at 90-degree angle, how they are giving the injection. So typically this injection forms a funnel type structure inside the skin. And this funnel dimensions actually will decide, the surface area of this funnel will decide the rate of release and your plasma drug concentration. Site of injection, like at the muscle site where you are doing the injection, that will decide. And the position of the syringe and how you are giving the injection will decide. So if you are using the syringe, keeping the syringe at 90-degree angle, then you will get a predictable result, better safety and efficacy. On the other hand, if you put it in an angular position, the needle, 30-degree, 60-degree, or anything between 90-degree to 0-degree, then the release profile will vary. Most of the times, ideally, it will become faster release, dose dumping will happen, and people will experience serious adverse effect and efficacy will be very low. Now, higher leuprolide acetate plasma levels are observed after injection to deltoid muscle arm than injection to lateral thigh. So if it is given into deltoid muscle, higher plasma drug concentration is achieved. Same injection. Again, higher leuprolide acetate concentrations are observed after injection to deltoid muscle and lateral thigh than to the buttocks. So in the buttocks, you will see the lowest plasma drug concentration, followed by lateral thigh and followed by deltoid muscle. Deltoid muscle, it will be the fastest release profile. Drug concentration will shoot up immediately. From the body also, very, very fast. On the other hand, it will be in the buttocks, it will be very slow. The reason very clear, in buttocks we have plenty of fat. So this the drug is also fatty in nature, all these fatty drugs, and they get dissolved in the fat and they give a very, very slower release profile when given into the buttocks, followed by the lateral thigh and followed by the deltoid muscle where the fat concentration is almost zero. So here you can see peak tryptophan acetate concentration in plasma after intramuscular injection at different sites of male and female volunteers. Here, earlier I talked about site of injection. Then I also talked about the positioning of the needle. Now I am talking about the plasma drug concentration, how it changes. The same injection, same site of administration, the same way of positioning the needle, but male versus female. So you can see the plasma drug concentration in female is coming slightly lower than the males. Why? Because in females, you have more adipose tissue, more fatty layer. In this fatty layer, the drugs gets entrapped and the drug concentration becomes low. Obviously, the efficacy also in females is very good as compared to males. All these long-acting injections, especially for reproductive tract disorders. Recent research suggests that the majority of the injections intended to be administered intramuscularly may actually be delivered into the fat. It has been found that less than 5% of women and less than 15% of men receive an intramuscular injection into the gluteal muscles when a 3.5 millimeter needle was used for injection. The majority of the IM injections may have been intralipomatous, that is into the lipidic tissue. Absorption of drugs from fat occur at a lower rate than the muscles or tissues, which I have already explained. Now, you people can correlate your experience. If you have administered the injection to somebody who is a bit bulky in nature, you will see his plasma drug concentration, low plasma drug concentration, and you will see him having a prolonged duration of action rather than people who is very skinny. They will be having a very, very shorter duration of action and adverse effects due to faster burst release. More superficial injection can avoid these complications and reduce the inter-subject variability. It is recommended that more superficial injection, so by reducing the length of the needle. But practically speaking, these injections, these drugs are highly variable. Their variability coefficient is very, very high. A needle of appropriate length should be chosen for the site of injection and for patients' deposit of fat. Now, improved adherence, greater tolerability, relapse prevention, fewer hospitalization, and reduced treatment discontinuation. These are the efficacies. So this is all quickly about the presentation which I had delivered. And as promised, I will share the presentation, a copy of this presentation to the organizers by the end of the day. I think now we can address questions.
Thank you, sir, for your kind words. Sir has highlighted about the long-acting injectable, especially suspension, microsphere, and in situ gel-forming implants and their application in the treatment of reproductive tract diseases. Thank you so much, sir. I hope you all are finding the national webinar valuable so far. We are now going to take a break for lunch. The break will last for 30 minutes and we will resume after 30 minutes and we'll have our second and third technical session by Dr. Vaibhav Dubey sir and Dr. Nilesh Mishra sir. So please be back by then. Thank you so much.
Thank you all. Thank you so much. Bye-bye.
Thank you, sir.
एक यूट्यूब का तो है तेरे पास आईडी पास और फार्मा क्लास सर ने मुझे से बाहर कर दिया हां मुझे पीपीटी वाले दे मुझे तो सर ने सबको तो बांटा है सबको तुम देखिए इस पर डाल देना हम किसी
तो मैम आवाज नहीं आ रही। सही। मैम एक्चुअली मुझे लग रहा है ना, यस सर।
फिर आप तू बोलती
तो ही करते हैं।
सर वो शायद यादव सर कहीं बिजी है, तो एक बार उनको फोन लगा लीजिए, अदरवाइज वो बोलेंगे तो फिर रिलीज जो है ना उनका करवा देंगे। ठीक है, हाँ ये मनीषा को बोला मेरे फोन लगा।
नहीं मांग रहा, भेज दो।
रहा है प्रश्न। नहीं आ रही, आ रही है थोड़ी थोड़ी। ये यहाँ पे ये क्या है? ये अपना वाला है इसका।
किसने भिजवाया ये कुछ ज्यादा गलत उंगली करते हैं यार।
और बोला है तो रजिस्ट्रेशन भी फोन आया तो बोला मंडे का बोलना।
कुछ देर में बोला कि वैशाली मैडम ऐसे आईटी जा रही है बता दो हमको इंजीनियरिंग जा रहे हैं अभी आएगी ना सर हाँ हम लोग जा रहे हैं उधर क्यों
I request all the participants to kindly mute your mic. Our second technical session is about to begin. I request all the participants to kindly be muted. We are shortly starting our second technical session.
welcome back to post-lunch session of National Webinar 2024 on Advancement of Nanotechnology in the Treatment of Reproductive Tract Diseases, a journey from bench to bedside, organized by Smriti College of Pharmaceutical Education Indore in association with Indian Society for the Study of Reproduction and Fertility. Moving ahead, we are very fortunate to have among us Dr. Nilesh Kumar Mehra sir, Assistant Professor, National Institute of Pharmaceutical Education and Research, Hyderabad. So I request Mrs. Manisha Dhare Madam to please display sir's profile slide. Dr. Nilesh Kumar Mehra sir is working as an Assistant Professor of Pharmaceutics at the Department of Pharmaceutics, National Institute of Pharmaceutical Education and Research, Hyderabad, India. He earned his PhD from Dr. H.S. Gode University, Sagar, and postdoc from Irma Lerma Rangel College of Pharmacy, Texas, USA. He served as manager in product development, Centis Research Center, Centis Pharma Private Limited, Gurgaon, for development, scale-up, and technology transfer of complex ophthalmic inhalation and optic pharmaceutical products. He received Team Award for successful commercialization of ophthalmic suspension product. He has authored more than 120 peer-reviewed publications in high-repute international journals and more than 30 book chapter contributions. He has granted 4 patents on manufacturing process and composition to improve therapeutic efficacy for topical delivery. He is guiding 14 PhD and guided 35 MS students for their dissertation research projects. He has received numerous outstanding awards, including Young Scientist and Team Awards for his research output. He is a peer reviewer of various international journals and publishers. He recently published 3 edited books on multifunctional nanocarriers, biopharmaceutical, and nanotechnology. He has rich research and teaching experience in the formulation and development of complex, innovative ophthalmic and injectable biopharmaceutical products, including micro and nanotechnologies for regulated market and so on. According to announcement by the Stanford University scientists based on the database and basis of standardized citation indicators, Dr. Mehra's name is included in top 2% scientists in the world in the area of pharmacology and pharmacy consecutively for 3 years. So, we welcome Sir for his insightful talk. Sir, now you can share your screen.